Hey guys, hi, welcome to Unacademy. This channel is Unacademy Future Doctors and I am your educator for biochemistry, Dr. Shivangi Shukla. I welcome every one of you here on this channel and we have been discussing MCQ based sessions. Okay, first of all, let me just check if the live stream has started and if I am visible and audible to everyone. Just give me a minute to check, okay? All right, just a minute. Just a minute, guys. You can also let me know if I'm visible and audible. If the stream would not have started, then your problem. That is why check in advance if the stream has started or not. Alright, I think the stream is going on fine. Let us just resume. Okay. So I am your educator for biochemistry. I am Dr. Shivangi Shukla and I welcome all of you to today's MCQ based discussion session. Okay. Now, I'll just quickly brief you about the different types of subscriptions for NEET PG that are available on the Unacademy app. Both Iconic as well as Plus subscriptions are available. Now, according to your needs, you can choose either of these. Both of these will provide you with access to video lectures from top educators across the country. Not just their video lectures, but also their PDF notes, the printed PDF notes, and question banks that are regularly updated containing more than 25,000 questions. Okay. Also, the USP of both of these subscriptions, Plus as well as Iconic, is that you can access these while on the go on any device of your choice, be it your phone, your laptop, your tablet, your PC, wherever. Okay, and you have the ease of access even to the recorded sessions. Okay, so and the Iconic subscription will also give you access to resources from Prep Ladder as well. Okay, so it is best from two of the best. All right. Yeah. So, and the special classes that Unacademy provides you with, the free special classes, they have state-of-the-art features like raise your hand feature, polls, in-class polls. So all of these ensure that the class is an interactive one. Also, Unacademy has also come up with uh, open house sessions. Okay, these open house sessions are interactive audio sessions, 1:1 sessions. With the educator, the learner can speak directly to the educator just as he would be speaking over the phone. Okay. Now, there's this revision batch from March 16th to May 16th, which is specially dedicated to those who are going to take this year's NEET PG examination. So there's a revision session where more than 40 educators will be taking revision lectures and grand tests subject-wise as well as full course tests. Okay, so you can enroll in this one. Also, while taking any of the subscriptions, do not forget to put in the educator code, that is DRSHUKLA. This code is going to fetch you an additional 10% discount on any of the two subscriptions. Okay, Dr. Shukla, DRSHUKLA. Do not forget this code. So we have completed a few of these questions yesterday. If you haven't followed, you can follow from the last session. Yeah. So what are we going to do today? We are going to start with this question. C-peptide is a component of which of the following? So I'll ask the question. You have to answer in the comment section within 2 minutes. I'll give you 2 minutes for answering in the comment section, and then we'll discuss. So C-peptide is a component of which of the following? Is it A, insulin, B, fibrinogen, C, proinsulin, or D, prothrombin? And your time to answer starts now. All the best. Come on. Come on, guys. Come on, answer this. Do let me know. Do let me know the answer, guys. Last few seconds left for you to answer. Come on, try and answer this. This one's easy. Alright. So the correct answer here is, it is proinsulin. Okay. Alright. Proinsulin. Now what is proinsulin? Proinsulin is the inactive form of insulin. Okay. It is the inactive form of insulin. Now, how is insulin secreted? The secretion of insulin, or the synthesis of insulin, occurs as a pre-pro molecule, that is pre-proinsulin. Okay. Now, what is pre group here? The pre group is the guiding group. Is the guide. Okay. This pre group, it will guide the proinsulin towards what? Towards processing. Now, proinsulin is what? proinsulin is the inactive form of insulin. Inactive or the precursor form of insulin. Now, why is insulin secreted like this? Because so that it is only released in the bloodstream whenever it is needed. Okay, unnecessarily, if it is whenever it is secreted and it is released as such, then what will happen? It will start acting. So therefore, the hormones or the enzymes that need to act only at a particular time are secreted. The inactive form, like coagulation factors, like collagen also, like parathormone also, PTH. Okay, now this group will guide the proinsulin towards the endoplasmic reticulum, where this proinsulin will undergo processing. Okay, this proinsulin will then undergo processing to produce insulin. And once the pre-group has guided the proinsulin towards the endoplasmic reticulum, the pre-group will then detach and go away. Okay, the pre-group will then detach and go away. Understood? Then what will happen? What is the structure of the proinsulin? See, I'll show you the structure of proinsulin here. This is proinsulin contains, there is this one chain that is B chain. Okay. Then there is another chain that is C-peptide. It's quite big. And then there is another chain that is A-peptide. Got it? These are the three chains. Now this one is B chain or B-peptide. This one is C-peptide. And this one here is the A-peptide. Got it? Now what happens is, during processing, what happens, there are nicks that are placed here so that the C-peptide is removed. So this one is the proinsulin. The C-peptide is cleaved out and there will be disulfide linkages between the cysteine residues of A and the B chain. So there will be disulfide linkages here. Cysteine is a sulfur containing amino acid. Cysteine and methionine, both of these are sulfur containing amino acids. Okay, so between these A and the B peptides, there will be disulfide linkages to link them, and this C peptide will be cleaved. Now this B and A chain containing molecule is what is called insulin, and it is functional. Now, what is the advantage of the C-peptide? Now, the molecular weight of C-peptide is almost similar to the molecular weight of insulin. The amount of, can you see how huge it is? So, to measure insulin, you can also, the insulin secreted inside the body, you can also measure the amount of C-peptide. Now, if someone is taking insulin from outside and you want to see how much insulin his body is secreting, it will be very difficult. Because if you measure the amount of insulin that is circulating in the blood, it can mean either the endogenous insulin or the exogenous insulin that is secreted. Okay, so what happens here is that you can, in such a case, you can measure the amount of C-peptide, because C-peptide will refer to the amount of endogenous insulin that is secreted, because it is equal to the amount of insulin that is secreted. Another case is to differentiate between type 1 and type 2. In type 1 diabetes mellitus, what happens is there is autoimmune destruction of the beta cells of the islets of Langerhans. So there is little or no insulin production right at the outset. So there will be very little C-peptide that is present, or maybe negligible or no amount of C-peptide that can be seen in blood. Whereas in type 2, what happens, the main pathology is peripheral insulin resistance. So because of peripheral insulin resistance, the peripheral cells, they cannot take up glucose because the GLUT4 receptors aren't expressed. The GLUT4 receptors are expressed in response to insulin in the blood. Whenever insulin is not present in the blood, because the glucose uptake in peripheral cells is insulin dependent. So the GLUT4 receptors will not be expressed, so glucose will not be taken in by the cells. So what will happen? There will be a signal of hyperglycemia in the blood in spite of insulin being present. Why is it happening? Because the cells are revolting against insulin. The cells don't want to acknowledge the presence of insulin. There is resistance to the presence of insulin in the peripheral cells. So what happens is, now the beta cells do not know that this is what is happening. So what will they think? They will get the signal that you're not working enough, you're not producing enough. So they will start producing increased amounts of insulin. Once they start producing increased amounts of insulin, there will be stress on the beta cells because they are working doubly, quadruply. Okay, they are working late at nights. So what will happen? Because of that stress, the beta cells are going to get exhausted and they're going to start dying. So the beta cell destruction occurs very late in type 2 diabetes. Initially, there is C-PEPTIDE, there is normal or increased amounts of insulin production, endogenous. So the C-PEPTIDE in type 2 diabetes mellitus can be normal, C-PEPTIDE levels can be normal or even increased, whereas in type 1 diabetes it will be negligible or no C-PEPTIDE levels. Understood this? So this is the utility of C-PEPTIDE, very, very important. Understood or not? Shall we move to the next question? Alright. Which of the following regarding glutamine is true? Glutamine, it is an amino acid. A, it transports ammonia. B, it cannot cross the blood-brain barrier. C, it is highly toxic. Or D, it is stored in the smooth muscles. Alright, the time to answer has started now. Do answer in the comment section. Come on guys, do let me know. Last few seconds left. Okay. So the correct answer here is, it transports ammonia. Okay. Now glutamine can cross the blood-brain barrier to remove the ammonia present in the brain. It is not toxic. What is toxic is ammonia. Ammonia is toxic, and that has to be conjugated with glutamine. It has to be coupled with glutamine in the blood for transportation to the liver, where ammonia will be converted to what? Ammonia will be converted to urea. And this thing is what confuses most people is, it is stored in the skeletal muscles. Glutamine is an amino acid that, along with other amino acids like glycine, they are stored in the skeletal muscles, and that is why the bodybuilders and all, they take glutamine, glycine supplements. Okay. You must have heard of BCAA, branch chain amino acids and all. Okay, so these are the supplements that bodybuilders take to build more muscular tissue, because skeletal muscle. Why? Because they are the constituents, they make up the proteins present in the muscles. Okay, understood. And why is it needed to transport ammonia? Because free ammonia is toxic. It can cause a lot of toxic symptoms in the blood. Therefore, it needs to be coupled with glutamine for transportation. And why is ammonia coupled with glutamine, and why is it there in the blood in the first place? Because it is a product of metabolism, especially protein degradation, and this ammonia needs to be converted to what? It needs to be converted to urea, where urea synthesis occurs in which organ? In which organ does urea synthesis occur? Inside the liver. And what are the organelles where urea synthesis occurs? It occurs in the mitochondria, and a part of it, the first two reactions occur inside the mitochondria, and the rest occur in the cytosol. Understood? Next question. Which of the following cannot be used for viral illnesses? A. Gram staining, B. ELISA, C. PCR, and D. Northern blotting. Okay? Your time to answer has started now. Okay? Okay, last few minutes left. Last minute left, come on. Which of the following cannot be used for viral illnesses? Viral illnesses means those illnesses where the pathogen is a viral agent. Now, what is special about viral agents? Come on, rack your brains and you'll know the answer. Last 10 seconds left. Okay, alright. So the correct answer here is A, gram staining. And why is it so? Viral illnesses, see, the special characteristic about the virus is that it just has its own genetic material. Okay, the nucleic acid, either RNA or DNA, single stranded or double stranded. Now, what it does is, for its own life cycle to run, the virus utilizes the machinery of the host. The host can be any organism. In bacteriophages, what are bacteriophages? Bacteriophages are those viruses that infect the bacteria. Bacteriophage, phage means eating. They eat up bacteria. That means they infect the bacteria. So for bacteriophages, the host is the bacteria. For human viruses, the host is the human cells. Okay, so depending upon the host cells, the viral nucleic acid will take up the host cell machinery to run its own life cycle. Therefore, what are viruses? They are the connecting links between living and non-living. Okay, because whenever they're outside the host cell, they're non-living. Once they enter the host cell, they start behaving as living organisms capable of reproduction. Okay, capable of carrying out their life functions, utilizing the machinery of the host. Now, to detect viruses in any illnesses for diagnostic purposes, to detect viruses, there can be either direct methods or indirect methods. Now, direct methods, the gold standard can be culture. If you see the viral particles, then it is the gold standard. Okay, nothing can beat that. But because viruses cannot multiply in vitro, that is outside the cell, because there is no host machinery. Okay, unlike bacteria, because bacteria are self-sufficient. They can multiply wherever you keep them in the culture plate. They just need nutrition and proper conditions. But for viruses, they also need a machinery. So the viral culture is a bit intriguing and a bit complicated. They require a lot of vero cell line cultures, chick cell embryo cultures. They are different complicated cultures. Okay, where the host cells are also there, and they are infected by viruses. Okay. Anyway, so this is viral culture, which is a bit complicated. Staining is typically done, gram staining is typically done for bacteria. Normal bacteria, typical, atypical, acid-fast bacteria, different types of staining. Now, gram staining is done where it distinguishes between gram positive and gram negative bacteria, depending upon whether a layer of mucopolysaccharide is present outside the cell wall or not. Okay, so this gram staining proper in detail you would be reading in microbiology in your second year. But this, at least this one you should know. Now, ELISA is an indirect method of detection of any disease. It can detect bacterial diseases also, viral diseases also, any kind of diseases it can detect. Indirect method means it will detect the antibodies to the agent. The antibodies that are present against the virus or against the bacteria or whatever you're targeting, that method is called ELISA. To any antigen, antibodies to any antigen. Okay. Now, with respect to COVID, the RAT test, that is the rapid antigen test that you do, it utilizes similar principle as ELISA. What is PCR? It is polymerase chain reaction. It is a direct method because it will amplify the viral nucleic acid, the viral DNA, and the viral RNA. It will first be converted to DNA. This I'm talking in case of with respect to COVID. The viral RNA will be converted to DNA, which will be amplified. This whole procedure we have covered in the clinical applications part. So you can go back and read and attend that session of PCR. Northern blotting, guys. Northern blotting is for RNA. So all the RNA viruses, their RNA can be detected using northern blotting. Now one more thing, southern blotting. What is southern blotting for? They are all immunosorbent assays, like paper chromatography. They are like that. Okay, now southern blotting is for DNA. Western blotting is for proteins. Understood? These three are very important techniques. Very good, Vashu. Very good. That is great, because biochemistry, these are the topics that you have read in chemistry and biology for a NEET preparation, isn't it? That is very good. Congratulate yourself. And you are in, just crack NEET UG and you will do very well in your first year at least. Okay. Now move on to the next question. I think, Vashu, this also you will be able to solve, because glycolysis, this is regarding glycolysis, this also we read in our PMT entrances, right? So all of the following are key glycolytic enzymes except A, glucokinase, B, glucose 6-phosphatase, C, pyruvate kinase, or D, phosphofructokinase. Come on, 2 minutes for you to answer. Last few seconds left. Come on. Last 10 seconds left for you to answer. Okay, so the correct answer here is D, glucose 6-phosphatase. Now, glucose 6-phosphatase is, what does this do? It will break down glucose 6-phosphate to glucose. And where is it used? It is used in cycles that are opposite to glycolysis. Glycolysis will use up glucose, whereas this is going to produce glucose. So it can be used, this enzyme comes into action in gluconeogenesis. Also in glycogenolysis, that is breakdown of glycogen. Okay, glucose 6-phosphatase. Glucokinase, what is this enzyme? It is hexokinase in the liver. Hexokinase is the enzyme that converts glucose to glucose 6-phosphate in all the cells. In the liver, it is known as glucokinase. What is pyruvate kinase? It converts pyruvate to lactate. This is also glycolysis. Phosphofructokinase. What does it do? Fructose 6-phosphate to fructose 1,6-biphosphate. Phosphofructo and kinase. Already there is one phosphate on the fructose, and kinase, it will transfer another phosphate. Okay, so it is phosphofructokinase. Understood? So I think we'll stop here, and we'll meet again in the next 2 sessions, and also at night at 11:45 we'll meet in the special class that is on the Unacademy app. That is an MBBS session, that is MCQ bonus biochemistry session, and there are 2 more YouTube MCQ based discussion sessions left. So you also please attend those sessions as well. Okay, alright then, see you guys, bye bye, take care. Do subscribe to this channel if you haven't for regular notifications of my classes, and also follow me on the Unacademy app. If you haven't downloaded the app, then do download the Unacademy Learners app. There, look for go to the NEET PG section, look for Shivangi Shukla, and you can follow me to get regular updates of all the classes that I schedule. Alright.